Bismillahirrahmanirrahim. 5090 Syllabus 2023 Specimen Paper 2 O-Level Biology. Now looking at the paper 2, the important thing which you've got to see in tip 2023, it says for examination from 2023, the timing has remained the same, 1 hour 45 minutes. But there's something important that you have to see is that answer all questions. Previously there used to be a section A, section B and a section C. Now there is no such thing as there are no choices and there are no section C's. You only have to do the entire one. There's no section. You have to do the entire paper and there are no choices. So this is the difference in 2023. The main difference you've got to realize is that there's going to be no section B, no section B which used to be essay questions, and no section C which used to be a choice. So no section B, no section C, it's all answer all the questions. Another thing you've got to realize is the total marks for this paper is 80. Now that is the same as previous exams. So 80 marks, that's a lot of marks for paper 2, and this is usually the most difficult paper that we are going to be talking about. The first question is digestion in the human digestive system is carried out by the action of enzymes. The diagrams each represent the action of a specific enzyme to break down a substrate into one or more end products. One or more end products. Read that carefully. Figure 1.1 has been completed for you. So protein was the substrate. This is a solved example. And amino acids were the end product. So the name of the enzyme was pepsin, and where the enzyme acts was stomach. So they've done this for you. They've done this question for you. I do not agree with the amino acids because actually here peptides would be formed. So amino acid is an ellipsin in the smaller intestine, but I mean I can't argue with Cambridge on their mark scheme. And this is a specimen paper. So then you have to complete this. Figure 1.2 was name of the enzyme, starch, maltose, and name of the enzyme here you've got to give. So somewhere they've given the name of the enzyme. So here naturally it would be amylase, and where the enzyme act would be in the mouth. But then you've also got an amylase in the small intestine, so you could have written duodenum or the small intestine as well. So any one of these was correct. When I put a slash here, I mean this is the alternative. You could have written mouth or small intestine. You don't have to write both. Then coming to lipase, then we have figure 1.3, then we have lipase. We've got something to write here, we've got something to write here, and you can see here they've written end products, so you know that what are the end products will be what. So you could have said fat, substrate is fat, and of course the end products are fatty acids. If you look at previous videos of biological molecules, that's what we studied. There are actually 3 fatty acids. I don't think you need to write that, and the other which you had to write was glycerol. Please do not write glycerine, or something like that because you usually mess this up. So where the enzyme acts, you know it's produced by the pancreas, so it's going to act in the duodenum, and this is what you have to understand where it acts, but you could have said small intestine as well. Here you could have said lipid, or you could have said oil, all were correct. Here duodenum, you could have said duodenum, or you could have said, even here you could have said small intestine. Because duodenum is the first part of the small intestine, so Cambridge is being kind to you and allowing you to be writing this. Here when I said 3 fatty acids, you don't have to write this 3, I'm just trying to make you revise it, and it's 1 glycerol. But you didn't have to write these, you just had to write fatty acids, or fatty acid was also correct, and glycerol was also correct. Then coming to the B part of the question, amino acids are the end products of protein digestion. Fine, so proteins to peptides, peptides to amino acids. Describe what happens to these amino acids after digestion until they reach the liver. And that's for 4 marks. Always look at the marks. So you must come up with 4 points. Now, when you write 4 points, I usually put these dots like this. Or you could write 6 points. Because even if you write something which is irrelevant, there's no negative marking for it. So if you've given me 4 correct points, well, I'll give you 4 out of 4. So this is how you learn to attempt the paper twos. The paper twos is write a lot, write more. You know, the lines don't tell you how much to write, but give more information. So maybe, you know, one of your information is slightly worded incorrectly and you don't get a mark, but for the rest, then you get the mark. So what happens to these amino acids after digestion? Naturally, what is going to happen to them? They're going to be absorbed. They're going to be absorbed by diffusion in the villi. They will enter the blood capillary, and then they will enter the blood plasma, and then they will be taken through the hepatic portal vein to the liver, because that's the intestine connects the liver, and the small intestine is connected by the hepatic portal vein. Now, as you can see, the answer to this was if you said, well, the amino acids are absorbed after digestion and then they have diffused into the blood. And where is this happening? This is happening in the villi, and they enter the capillary. Why? Because the capillary is one cell thick, and of course, arteries and arterioles would not allow it to enter, or venule or veins would not allow it to enter because they are thick walls. So they will enter the capillaries, and then where do they enter the blood? Blood plasma. You see, the thing is that when oxygen is transported, it is transported as oxyhemoglobin inside the red blood cell. So blood is a tissue. There is the plasma in it, and then there is the cells in it. So the red blood cells and the white blood cells. So you have to be very clear when you're going to talk about the plasma and when are you going to talk about the things being transported in the cells. And then the vessel which connects is, of course, is the hepatic portal vein. Now you can look at it. There are 1, 2, 3, 4, 5, 6 points, and there are only 4 marks. So any 4 out of these. I'm not saying you have to write all of these to get the 4 marks. Any 4 out of these, and you got your 4 out of 4. Now coming to question number 2, figure 2.1 shows the effect on crop yield amount harvested of using nitrogen containing fertilizer. Now you can see crop yield is kilogram per 10,000 meters square. So it is the area which is important. So we're going to talk about, this is the area 10,000 meters square. How much crop did we get? How many kilograms of wheat did we have? How many kilograms of sugarcane did we have? I have a farm, and you know, we always do our sums is how much wheat crop did we have? How much rice crop did we have? So crop yield is in kilograms, mass per unit area, per 10,000 meter square area. Now the mass of the fertilizer is kilogram per 10,000 meter square. So how much fertilizer did we add in this 10,000 meter square area, and how did it affect the crop yield, the mass of the crop that we got? How much sugarcane did we get? How many mangoes did we get? How much wheat did we get? How much rice did we get? Now it says use the information in figure 2.1, describe the effect. Now I always tell you this, if you've looked at my other videos, describe is no biology. It's no biology. You just have to give me substances from the graph. You have to just tell me, okay, what does the graph tell you? So the graph is telling you at 0, this is about nearly 800. And then as we've increased the fertilizer to 50 and 100 and 150, it has increased. But then when we've increased to 200 and 250, it has leveled out. So it's constant. So the crop yield has not increased. It has remained constant. I'm not saying it's not there, it's there, but it has remained constant. So when it says describe, I don't want any biology here that, oh, adding nitrogen adds what, and the glucose and nitrogen combine to form amino acids. So nitrogen is the limiting factor, more the nitrogen in the fertilizer, more the protein. So I'm not going to, I said no biology. So these 3 marks are marks which you should get, everybody should get, because actually if you don't know any biology, you can still do this part of the question. So you wrote this, no fertilizer, 0. Yield is 800, but you have to write the units as well. Units is kilogram per 10,000 meter square. This is very important that you write the units. We are we are okay with it in O levels, but in A levels we don't give you a mark if you have not written the units, whatever the units are given on the graph. In describe, you have to describe the graph. So the first point you wrote is no fertilizer, yield is 800. So you talked about the 0 part. But then as you've increased it by 50, you've added for 50 kilogram of fertilizer. As fertilizer added, increased crop yield. So the fact that there is an increase in the crop yield, you've explained that point. Then you've given me the highest point. So in 150 kilogram, the yield was this, 5,600 kilograms per 10,000 meters square. Right? Now the last point is high fertilizer. This is above 150. There is no increase in crop yield. because here what you see, it has become constant. So that means the crop yield is remaining the same, 5,600. So the crop yield has not increased. So this will be the last point, will be high fertilizer, or you can say above 150, no increase in crop yield. So the last point I've given it to you is above 150, no increase in crop yield, which was of course explaining this part of the graph, in which of course the level has remained the same, this part of the graph, in which we have increased the the fertilizer from 150 to 200 and then to 250, but the crop yield has remained the same. Now coming to part 2 of the question, the nitrogen in the fertilizer is in the form of nitrate ions. Fertilizer is in the form of nitrate ions, that we all know that. Describe how the nitrogen in the fertilizer is absorbed by crop plants. Number one, how is it absorbed and used to give an increase in crop yield? Why would it increase the crop yield? Now that's very simple. Where is it absorbed? It's absorbed in the root hair. You can't just say root. So the root hair cells, that's where it is absorbed, and this is absorbed by active transport. So look at the marks of it. The marks were 3. So you said absorbed in the roots. Well, I can't give you a mark, but you said active transport, or you said against the concentration gradient, or you said down the concentration gradient. All will get you this 1 mark for active transport because you're explaining it. And what is the nitrogen used for? It is used for the production of proteins. You know the nitrates are combined with glucose and form amino acids. So production of amino acids. So the glucose and the nitrate combine. You can see this in a previous video of mine. So production of amino acids, or you could have said production of proteins. Basically, protein are made up of amino acids. And this, of course, more the protein means more increased growth. Just like even you need more proteins when you're growing up in your teens. So more proteins means more growth. So how did we get more crop yield, more wheat, more sugarcane, more rice? So basically, this was the 4 points that you had to give me, but you could have given me maybe this point, and this point, and it would be fine. I would give you your 3 out of 3. Then coming to part 3 of the question, explain why a farmer may decide to use a mass of fertilizer per 10,000 meters square, which is less than that needed for a maximum crop yield. Now, this is a little difficult question. Why would a person use less fertilizer? Why? Because you see, when you add the fertilizer, most of it is not absorbed by the plant. So what is absorbed is fine, but what is not absorbed is going to be leached out, is going to be washed by rain into lakes and rivers and ponds, and then when it goes into lakes and ponds, is going to be eutrophication. And when there's going to be eutrophication, there's harm to animals, or the fish are going to start to die. Moreover, the fertilizer costs a lot of money, so it could be a lot of high cost, or it could be very, very expensive. And the second thing is, this possible economic return is not beneficial over the increased cost, because if you spend say 10,000 rupees and your crop didn't sell for 10,000 rupees, well, the cost of the fertilizer is a waste. So you have to think of the economic benefit. If you're not going to get that much benefit, then why add so much fertilizer? Fertilizer costs a lot of money. So as you can see, I've written the points which was supposed to be given by you, and you can see there are 5 marks in point, but there are only 3 marks. So runoff, or the nitrates are washed away by rain, which is called leaching. Now the word is leaching, Leaching, leaching. You must know these technical words. And this will result in eutrophication, or you could have explained the process of eutrophication, even then you got a mark for that. And this would result in harm to animals and high cost slash expensive, and possible economic return not beneficial over the increased cost. Then coming to the last part of the question, state one type of mineral other than nitrate that is required by a plant and explain. So type of mineral oil is magnesium, and this is in your syllabus actually, magnesium. And why do we need it? We need it for the importance to plant is we need it for the chlorophyll molecule. Please do not say chloroplast. We need it for the chlorophyll molecule, which is of course going to trap light and is going to be the important for so light energy to chemical energy, and it is needed for photosynthesis. So basically, chlorophyll is needed for photosynthesis, and that is, you could have got a mark for that as well. If you didn't say chlorophyll, you could have said photosynthesis, or you could have even said traps light energy. That completes this question, and this was for 11 marks. Now coming to question number 3, DNA controls cell function by controlling the production of proteins. This is of course in the new syllabus, so of course we will do this in the last chapter. These proteins determine the phenotype of the organism. What color of skin are you have, what color of eyes will you have, what color of your hair will you have. Phenotype, the physical characteristics of an organism, including blood groups in humans. Describe the structure of a DNA molecule. It has 4 marks. Now, if you've done the structure, you've done it in biological molecules. It's 2 strands, it's a double helix, it is made up of nucleotides, and the nucleotides are adenine, thymine, guanine, and cytosine. You could have just said A, T, G, C, and then there are base pairs, and then there are bonds between the bases, and the bases always pair up in the same way. So you can see what are the things: 2 strands, double helix, DNA is made up of nucleotides, they are A, T, G, C, and they're always base pairs, and the bonds between the bases, actually the hydrogen bonds between the bases, and the bases always pair up the same way. You know, A always pairs up with T, and G always pairs up with C. It could be like this, could be T here and A here, or C here and G here. So they will always pair up in the same way. There's no sort of anomaly in it. It will always be the same, right? So this is question 3 part A, which I have solved. B part of the question: one parent of a child has blood group B and the other parent has blood group A. The child has blood group O. The parents decide to have another child. Complete the genetic diagram to show the possible blood groups for the second child of these parents. So genotypes of parents and gametes. So possible genotype of child and possible blood groups of the child. And this is 4 marks. So how do you do these genetics question is you've got to understand is that what did they tell you in the question? They told you in the question that the child, one parent of a child has, so one parent is group B and one parent is group A, but the child is O. So if you have to have IO, then the IO has to come from each parent. Only then you can get an IO. But then we said this was parent was B and this parent was A. So if it is B, so this has to be IB and this has to be IA. This is how you do the question. You started from the answer. Like in math, sometimes you know the answer and you can do the reverse question. But here in genetics, if you know the child is this, so then you know it has to come, each allele has to come from one of the parents. So IO has to come from the mother and IO has to come from the dad. Now let's look at the other possibility. The other possibility, I'm going to use another color for it, IO and IA. So this is going to be IAIO. The blood group of the child in this situation will be O group, which is the possible blood groups of the child, because IO is the genotype, phenotype will be O group. This child will be A group. Now the next one, let's use another color for that. So IBIO, this person will be blood group B. And then we have another, the other one will be, so we have an O, we have an A, and but then we can also have this IB and IA. So IAIB, and this child will be AB. So there is a 25% chance of having any of the blood groups. So the question they've asked you is first complete the genetic diagram. This was for 4 marks. So what are the 4 marks for? Well, the 4 marks will be, this genetics questions are marked per line. So if you got this line correct, 1 mark. If you got this line correct, 1 mark. This line correct, 1 mark. And this line correct, 1 mark. So genetics questions are like maths. You can get full marks in them, but you've got to get it right. The next part of the question is part 2. State the probability that the second child of these parents has the same sex as the first child. Now you know that XX, XY, this story we've done in previous videos. So XX, XY. So there is a 50-50 chance always of having a male child or a female child. So this is 50%. State the probability 50%, or you could have said 0.5, or you could have said 1 in 2, or 2 in 4, or 1:1 ratio. So any of these. Now, has the same blood group as the first child. Now, if you look at the previous one, there's a 25% chance of O, A, B, and AB. 25, because 100% is the whole thing. So 25% this, 25% this, 25% this, and 25%. This is actually this is a syllabus point which they are examining from you. So the blood group having the same is 25%, or you could have said a quarter probability, or you could have said 1/4, or you could have said 0.25, 1 in 4, 1:3. So these are the all possible answers which you could have given in this question. Question 4, figure 4.1 shows a photo micrograph of a pollen grain with a pollen tube growing from it. A student placed pollen grains from the same type of plant in sucrose solutions of different concentrations for a fixed amount of time. After this time, the student used a microscope to examine the grains and tubes. Always read the question very slowly and very carefully. Table 4.1 shows the results of this investigation. So we have a 1% sucrose. Now always explain this to yourself. You have a 1% sucrose and the percentage of pollen germinated. I mean, if there were 100, only 6 germinated. Percentage of pollen grains germinated. And the mean pollen tube length was 0.005 millimeter. In the 2% sucrose solution, 13% out of 113 germinated. So we must have done separate experiments because we must have placed them in separate concentrations. And the mean length was 0.008. So it has increased the mean length. Look at it very carefully. In 4%, 25 germinated and the mean length was 0.015 millimeter. Always look at the units. In 8% sucrose, germination was 56%. That is the highest. And then after that, you see they are decreasing. Always look at a pattern. So 10, 20, 40%, they are decreasing the percentage of pollen grains germinated. And look at the length. The length increased to 0.040, and after that, the length is again decreasing. So length was increasing from here to here, the length was increasing. And then it's decreasing. So a total of 12 pollen grains were placed in 20% sucrose solution. Using the information in table 4.1, always look at it. Sometimes you look at the wrong one. Sometimes figure 4.1 and table 4.1. Be careful in that as well. One of the students on your YouTube has commented that, Miss, could you please help us to solve the paper 2s? So you must understand, when I am explaining these to you, I expect you to make notes of this and remember these things when you're doing a paper 2. A total of 12 pollen grains are placed in 20% sucrose. Using the information table 4.1, calculate the number of pollen grains that germinated to produce a pollen tube in the 20% sucrose. Let's look at the 20% sucrose. Here it is. 25% germinated. Look at the unit. 25% germinated. So how many will have germinated out of 12 pollen grains? 3. 25% of 12, half is 6. 25, so 50% would be 6. 25%, I mean, your maths is very poor, and if you're doing add maths or mad maths, well, you need to be a little more clear on this. This is a very simple sort of an answer, but if you get this wrong, you need to be something. I won't use the word which I would like to use the word here. Suggest why the mean pollen tube length was calculated for each sucrose solution. Mean was calculated. Why was the mean calculated? Because some may not germinate, some may not produce a pollen tube, or you can say to have a valid comparison. Like if I take the mean result of the test which I gave yesterday to the students. So some students got 50%, some got even 70%, some got even 30%, and some pathetic ones got 10%. So if I have to calculate the mean, then I would add all of them and divide it by the number of students. So suggest why the mean pollen tube was calculated. Mean is a very good way of comparison. So to enable a valid comparison, I take a test in school A and then I take a test in school B. And I say, okay, well, the mean in school A was this and the mean in school B was this. And of course, if the mean was less, then those students need to really do something to improve their skills in answering questions or improving their study skills. So suggest why the mean pollen tube was calculated for each sucrose solution. So some may not produce a pollen tube or to enable a valid comparison. Whenever you take out a percentage or whenever you take out a mean, the percentage also helps us to understand a valid comparison. If I have only 10 students in school A and I have 40 students in school B, then to take out a percentage would be a valid comparison. If I say 20% got an A and in the other school 40% got an A, well, you don't know the number of the students in the school, but you get a rough idea when I give you the percentage. So many percent of my students got an A in this school. So many percent of my students got an A. In one school I have only 10 students, in one school I have 40 students. So it's a very valid way of comparing it if I give you a percentage rather than saying, okay, 2 students of mine got an A. But if I only have 4 students and 2 got an A, but if I have 40 students and 2 got an A, so the percentage helps you to make a valid comparison. Then part 3 of the question, using the information in table 4.1, I defined the optimum concentration of sucrose solution of pollen grains germinating. Let's look at this. Optimum here, the highest was this, so this would be 8%. So, but the percent is written. Sometimes if they didn't write the percentage, then this would have been for 2 marks. So one would be for 8 and one would be for the units, but the percentage is written here, so they expected you to just write that. Then explain how you use the information in table 4.1 to answer A3. Why? How did you do that? Well, you saw the highest percentage of germination and you saw the longest length of the pollen tube. So highest percentage and highest length of the pollen tube. So highest percentage and longest length of pollen tube. This is what you would write. Now coming to part 5 of the question. The germination of pollen grain requires the movement of water into the pollen grain to form a pollen tube. So water has to enter the pollen grain. Now suggest by placing a pollen grain in a solution with a higher sucrose concentration than in your answer to A3 may result in a lower percentage germination. You see, this is the pollen grain. Water has to enter. But if inside you have if you have a 10% sucrose solution, then water will not enter because water enters by osmosis down the water potential gradient. So no water will enter because outside we have a very high concentration. So suggest by placing a pollen grain in a solution with a higher sucrose than in your answer to A3, 8% we said was the best. But if we put it in a 10%, so what will happen? In 10%, 8% water was entering and the germination was good. But if it is 10%, what is going to happen? Water is probably going to leave it and there is not going to be any, because it says the movement of water to form a pollen tube. Germination means to formation of a pollen tube. So no pollen tube formation is going to take place. So water enters by osmosis. So osmosis occurs down the water potential gradient, 1 mark, second mark. If you place it in a higher concentration, water will leave the pollen grain. 3 part, describe the route taken by a growing pollen tube in a plant and explain the importance of this route in plant reproduction. So describe the root, stigma, style, ovary. 1 mark, stigma, style, ovary or ovule or embryo sac, and the male gamete nucleus moves through this pollen tube, and ultimately fertilization is going to take place or fusion of gametes is going to take place, and of course an embryo is going to be formed and then the seed is going to be formed. That ends this video, and thank you very much. The specimen for the specimen paper 2 was discussed here. This is the first video, and another video will follow because I was not able to complete it in this video. So the rest of the questions will go into the videos 2 of this series.